Hello everyone and welcome. In this video, we're going to talk about the CRISPR-Cas9 system and its application to treat sickle cell disease. To start, let's ask ourselves, what is CRISPR? CRISPR itself is an acronym that stands for Clustered Regularly Interspaced Short Palindromic Repeats. This description of the acronym is essentially explaining the structure of CRISPR as short repeated sequences of DNA with spacers connecting them. This describes the natural state the system was found in, as was discovered as a mechanism in the bacteria genome to combat viruses by copying and incorporating a part of the viral genome to its own. This discovery was made by Dr. Emmanuel Charpentier and Dr. Jennifer Doudna in 2012. Scientists have then used this discovery of the virus-fighting genome in bacteria to derive the CRISPR-Cas9 system that is used as a hallmark for modern gene editing. To further understand the system, let's talk about its structure. The CRISPR-Cas9 system is a complex made of two components. The first component is a synthesized guide RNA, also sometimes referred to as gRNA. The primary purpose of the guide RNA is to identify the targeted DNA and guide the system to it. This guide RNA is a single strand that is complementary to the targeted DNA. The second component is a Cas9 endonuclease. The primary purpose of the Cas9 is to cut the targeted DNA once the guide RNA has located it, creating a double-strand break, or DSB for short, that allow for modifications in the genome to occur. These two components come together to form the CRISPR-Cas9 system that essentially acts as an RNA-guided DNA cleavage system, which is essentially a pair of scissors for genes. We talk about DNA and RNA heavily in this video, so if you're not familiar with these nucleic acids, we have attached an infographic in the description below for better understanding. Now that we have familiarized ourselves with the system, let's talk about how it works. Before the system can start operating, the targeted DNA must be identified first. Once the targeted DNA is identified, a guide RNA is synthesized to be complementary to a targeted DNA sequence. With the synthesized guide RNA and Cas9 endonuclease, a specialized CRISPR-Cas9 complex is formed and inserted in the targeted genome. The complex then looks for the targeted DNA by looking for a binding site called the protospacer adjacent motif, or PAM for short, which is a short DNA sequence downstream of the 3' end of the site of the targeted DNA in the genome. Once found, the CRISPR-Cas9 complex can initiate binding to PAM. Once bound, the Cas9 endonuclease cleaves the targeted region, creating a double-strand break. Then, the complex detaches from the genome as the targeted DNA has been cut and a double-strand break has formed. Following that, a repair mechanism occurs in two ways to mend the double-strand break. The first repair mechanism is called non-homologous end joining, or NHEJ for short. In this repair mechanism, the cell is left to mend the ends of the DNA either by joining the genome or inserting a sequence as a replacement for the cut targeted gene. This mechanism is referred to as non-homologous because the break ends are directly ligated without the need for a homologous template. The second repair mechanism is called homology-directed repair, or HDR for short. In this repair mechanism, a third piece of component is attached to the CRISPR-Cas9 system that acts as our DNA repair template. Subsequently, the gap DNA undergoes base pairing with the complementary ends of the repair template, and then DNA synthesis elongates the new synthesized DNA and closes the gaps. This results in a restored but genetically altered targeted strand. Following the end of either of the repair mechanisms, gene editing by the use of the CRISPR-Cas9 system ends, marking a genetically altered genome. It is worth noting that while genome editing is the primary use and function of the CRISPR-Cas9 system, the complex can sometimes be modified and then used to express the targeted gene rather than cleave it. This technique is used to study the targeted gene's function. However, this is a topic we will not dive deep into, as the primary focus of the CRISPR-Cas9 system is genome editing. Now that we know what CRISPR is and how it works, what is the significance of it? The most significant aspect of the technology is its convenience in laboratorical procedures. To start, the system provides a relatively inexpensive method for gene editing. This makes it very accessible and easy to perform tests with. It is also highly adjustable and specialized, thus it can be used to alter any nucleotide sequence in any cell, making it very versatile. In addition, because of its requirement for PAM as the binding site, each complex targets a specific DNA sequence and its corresponding PAM, allowing for multiple gene targeting events within the same cell to occur without any crossing. Lastly, the system requires only a synthesized guide RNA, and scientists use PAM as the specificity determining element for DSPs, also called double-strand breaks, making it a very simple method compared to other genome editing methods. Due to its programmable nature and minimal requirements, many applications of the CRISPR-Cas9 system have been derived. Some of these applications include rapid generation of cellular and animal models, functional genomic screen, and treating genetic disorders. The application of treating genetic diseases have provided huge advancements of treating human genetic disorders such as HIV, cystic fibrosis, Wolfram syndrome, and sickle cell disease. While all of these applications are significant, we're going to focus on the CRISPR-Cas9 system as a treatment for sickle cell disease to showcase the significance of the system. We will start by introducing what is sickle cell disease. Sickle cell disease, or SCD for short, is a group of blood disorders that arise from a point mutation in the hemoglobin beta gene. This point mutation occurs as a substitution mutation. We would like to know if you are unfamiliar with the topic of mutations. We have attached an infographic below to explain what they are and how they occur. The substitution mutation that occurs in the genome causes an adenine nucleotide to be substituted for a thymine nucleotide, altering the DNA sequence. Subsequently, this substitution causes glutamic acid to be replaced by valine, as the mutated sequence now codes for a different amino acid. Ultimately, this results in a different protein being synthesized called hemoglobin S instead of hemoglobin A during protein synthesis. This produced hemoglobin S makes affected red blood cells sickle cells. The name sickle cell comes from the shape the protein causes. Normal blood cells are round, whereas blood cells affected by sickle cells are shaped like a sickle or crescent moon. This change in shape of red blood cells restricts the cell's ability to flow fluidly throughout veins and arteries, meaning red blood cells cannot carry oxygen to organs and tissues throughout our body. This makes the disease extremely dangerous, as patients can feel weak and tired as the organs are not receiving a sufficient amount of oxygen. Now, with our introduction to sickle cell, we can talk about the CRISPR-Cas9 system as a treatment. As we have previously established in the workings of the system, scientists start by synthesizing a guide RNA to target the point mutation occurring in the hemoglobin beta gene. Once the guide RNA is synthesized and the CRISPR-Cas9 system is formed, the complex is inserted into the subject's genome in vitro, meaning outside of the cell. The CRISPR-Cas9 complex identifies the targeted mutation through binding to PAM and induces a break called a gene lock at this site to provide access to the targeted DNA. With this break, the point mutation is cleaved off. Subsequently, the genome repairs through HDR, as the CRISPR-Cas9 complex provides a donor template off which the cell will synthesize the correct complementary strand without the harmful mutation. Ultimately, the point mutation has been corrected and the genome is returned to a normal, healthy state. Now that the genome is corrected by the use of the CRISPR-Cas9 system, what are the results? Well, since the DNA is repaired, it is ready to be reintegrated in the cell. Once the edited genome is reintegrated, the cell can once again synthesize the correct hemoglobin A protein. This then ultimately produces spherical red blood cells instead of crescent-shaped ones, as the cell does not produce hemoglobin S anymore. With the constant production of hemoglobin A, the frequency of hemoglobin S is reduced and cycled out. Once the frequency of hemoglobin S is minimal, sickling of blood cells is terminated and red blood cells are once again healthy and round. We would like to know if you are unfamiliar with protein synthesis and how DNA sequence affects protein structure. We have attached an infographic regarding central dogma to expand and provide more information regarding the topic. Now that we understand the treatment, what stage is it at right now? Most completed trials have been preclinical and have only been conducted in mice test subjects. While mice are a good test subject to imitate the human blood system, they do not fully replicate it, which makes the technology yet to fully apply to human subjects. Clinical trials have just emerged, with the first clinical patient, Victoria Gray, being the first fully healed patient by the use of the CRISPR-Cas9 system. Thanks to the CRISPR-Cas9 system, her blood cells were terminated of sickle cell disease and are healthy once again. Gray is now free from complications that have taken over her life since birth. However, with that being said, the treatment is a long way from being FDA approved, as it is still new. In addition, scientists are still encountering challenges with the treatment that still need to be resolved. The usage of CRISPR to treat sickle cell disease has given rise to a harmful side effect in which insertion-deletion mutations occur within the targeted gene. Subsequently, this mutation contributes to the observed reduction in total hemoglobin levels in cells post-treatment. In addition to these challenges, CRISPR-Cas9 system itself possesses additional challenges. The first challenge scientists are constantly encountering are off and on-target effects. Off-target effects refer to the issues the CRISPR-Cas9 system encounters with targeting the loci on the target DNA. This mainly arises from the system's dependency to locate and bind to PAM, as the system can sometimes target undesired genomic loci. Subsequently, the system edits an untargeted sequence of the genome, resulting in unintentional changes, hence the name off-target. Off-target effects can lead to mutations, toxicity, and editing inefficiency. This poses a great challenge to scientists, as cells can be negatively impacted by off-target effects. On-target effects refer to the undesired outcomes coming from double-strand breaks created by Cas9. In this challenge, while the complex does target the proper DNA sequence and cleaves it off, the created double-strand breaks sometimes result in unexpectedly long deletions, causing more complex genomic rearrangements at the targeted site that potentially lead to pathogenic consequences. Another challenge with the CRISPR-Cas9 system is issues with the delivery of the system. One disadvantage with the CRISPR-Cas9 complex is its size. The Cas9 protein can contain 4 to 7 kilobytes of information. However, current delivery methods are only limited to around 4.7 kilobytes of DNA. Therefore, it is difficult to construct a virus that can encode such a large gene, its promoter, and guide RNA. In addition, in order for the CRISPR-Cas9 system to be successful, it must have effective cell targeting, rapid clearance after the CRISPR system works, and minimal cytotoxicity. However, it is difficult to simultaneously achieve all of these criteria with current delivery methods. A third challenge arises from the repair mechanisms that occur as a result of the CRISPR-Cas9 system. To start, the non-homologous end joining repair mechanism process is error-prone and relatively imprecise. As the cell is left to repair the edited genome, there is a high chance of insertion or deletion mutations of the DNA sequence if the cell does not produce the proper nucleotide sequence. These mutations hinder efficiency and can cause harmful unwarranted effects to the edited cell. While HDR is more precise and less error-prone than non-homologous end joining, this repair mechanism still possesses issues. The efficiency of HDR is comparatively sluggish, and this is the case because the mechanism depends on both homology arm length and DNA template type. In addition to this, HDR usually operates during the S/G2 phase in somatic cells, which makes it less flexible to work with. Another challenge that occurs in mammalian cells is that the system can trigger the immune system in mammals. This arises because the technology is derived from a bacteria, thus naturally the immune system of mammals can be triggered as a result when it recognizes the system. While the CRISPR-Cas9 system clearly has its flaws, it is undoubtedly a groundbreaking technology that has revolutionized gene editing. Thus, the future for CRISPR is very promising. Scientists are constantly developing the CRISPR-Cas9 system with new techniques and improvements emerging. This proves that scientists are not only invested in the system, but it is the present and future for genome editing. Scientists around the world are also working to combat the challenges we have mentioned in this video in order to make the technology more effective. Several methods are being developed to mitigate the off-target effects that can occur because of the CRISPR-Cas9 system. Specifically, scientists are trying to detect the off-target cleavages in order to select the single guide RNA with the lowest amount of off-target effects. In addition, scientists are studying the various delivery methods and comparing their pros and cons to optimize efficiency and reduce negative effects. Lastly, scientists are trying to combat challenges with the repair mechanisms caused by the CRISPR-Cas9 system. They are doing this by coming up with approaches to inhibit the key enzymes of non-homologous end-joining mechanisms using certain molecules. Combating these challenges will not only improve the system's efficiency, but make it more safer and effective as a technology. With such improvements come new applications that can revolutionize technology. These applications involve medicine, diagnostics, agriculture, synthetic biology, and treating cancer. However, it is important to note that an ethical issue arises from the CRISPR-Cas9 system. All the information covered in this video thus far have been surrounding somatic cells. Thus, the ethical issue arises when we start talking about germline cells, which are cells used in reproduction. The genetic editing of germline cells can result in DNA editing that affects an offspring's lives from start to end, without their consent. In addition, scientists can edit the genome of an infant with the same impact. This makes it evident that baby engineering is a possibility. However, a concerning one, as permanent changes like this are uncharted and unethical. For that reason, scientists around the world have been taking a strong stance against such applications, as 75 countries have prohibited the use of CRISPR-Cas9 in human reproduction. To leave you with a parting thought, we wanted to share a quote by the co-founder of the technology, Dr. Jennifer Doudna. The quote says, "The more we know, the more we realize there is to know." It is amazing how the technology that started as a curiosity by a couple of scientists transformed into a tool used to change human lives. Thus, the CRISPR-Cas9 system does not only stand as a pillar for cutting-edge technology that can revolutionize human society, but as an example of what can be followed by curiosity and research. Thank you for listening, and we hope you enjoyed the topic as much as we have.